All right, so as Angela mentioned, I'm Phoebe Zarnetske, and I run the Spatial and Community Ecology Lab here at MSU. And we focus on trying to understand patterns of biodiversity and how species interact with each other and how climate change in particular might affect those. And Elise?
I'm Elise Zipkin. I run the Quantitative Ecology Lab, also in the Department of Integrative Biology. And my lab focuses on understanding the abundance and distributions of species and the factors that affect those across space and time. And you'll see that we work on a lot of different taxa, but our specialty is really the quantitative aspect of it, developing models, and we work closely with biologists who are experts in each of those different taxa groups.
All right, and we'll return to this slide at the end. So if you want to check out our, excuse me, our websites, you can feel free to do that. So as we get started here, we wanted to talk a little bit about biodiversity, what really biodiversity is. A lot of people think about biodiversity as being the number of species at a given place and time, and that's correct, but there's also a lot of other dimensions of diversity to consider. Here's just a map of the portion of Central and South America showing where biodiversity is high and where it's low as quantified by the number of species. And that differs depending on birds, mammals, and amphibians in this case. So a broader definition of biodiversity is really the variation of life on Earth. There can be multiple dimensions of biodiversity. This can be the diversity within species, so genetic diversity, or the diversity of different characteristics of species and their populations. It can be the numbers of species, but it can also be the numbers of unique ecosystems as well. We know that biodiversity is essential, and unfortunately, we're losing it really fast. A recent report compiled by experts, biodiversity scientists around the world, estimates that there's 1 million species at risk of extinction, and that's out of about 8 million species that are known. Scientists believe that there's could be several more million species that haven't even been quantified yet or discovered. So this could be a low estimate. And biodiversity is essential because it provides us with a lot of resources. Many of us like to go for hikes and enjoy the outdoors. That is essentially a lot of biodiversity that's providing that service. Also public health, there's a lot of diseases that biodiversity can help with in terms of medicine. And of course, biodiversity provides food that we all rely upon. And so it's providing a lot of important services. But there are a lot of threats as well. And those threats can impact different taxonomic groups. And all major taxonomic groups are being affected by these threats. For example, amphibians have about a 40% decline in terms of the risk of extinction. Conifers, 34%. And so these different taxonomic groups have different levels of threats, but you can see that they're all being impacted. And those impacts are happening more and more as we go through time. So as the human population grows and there's increasing threats upon biodiversity, it's just really increasing. And this is just a plot showing the various different taxonomic groups and how over recent history, the number of endangered species, so the classification of the most extreme risk, is increasing every year. So what are some of the causes of biodiversity loss? Well, that same report identified these as the major causes. Climate change is certainly a really important one, and it can interact especially with habitat degradation. So clearing of land, essentially for potentially urbanization or growing agriculture. And that can also have a lot of feedbacks that affect the fire frequency. For example, this is an image of the recent fires in the Brazil Amazon, which is really a combination of land use change and climate change impacts. Biodiversity is also threatened by direct exploitation. So overfishing, for example, or poaching, and pollution, disease, and invasive species are other threats as well. Importantly, these threats don't have a singular effect on any given species. They're really a combination, and so they have many synergistic effects. When we think about where the threats are highest for biodiversity, we can actually start to quantify that around the world. And this is a map of biodiversity hotspots around the world. And these are areas where there's a combination of high risk to those species and the levels of biodiversity, but it's also areas where we have the highest degree of endemic species, or species that are native to that region. So they're both high diversity areas and they're high risk. And these areas have been a real focal point for conservation because they are at such a high risk. Unfortunately, they also coincide with areas that we know probably the least about. We know a lot more about North America and Australia, for example, than we do about a lot of the tropics, where diversity is the highest. So a lot of research needs to be done more in those regions. So climate change is probably our biggest grand challenge right now. And as I mentioned, it does interact with a lot of these other threats to biodiversity. This is a map or a diagram just showing the global mean temperature as indicated by different color bars going from blue, cooler, to redder colors being warmer. And it goes from 1850 to 2020 today and over into the future to 2200. And what's shown on the right are these different potential scenarios, depending on what we as a society decide to do in terms of curbing our emissions. So we could be seeing a future that has a very extreme temperature level if we don't curb our emissions and cut global warming, reduce global warming. Or we could decide to make some decisions that would actually curb those emissions more effectively and keep our temperature below 1.5 degrees, which is the expected kind of range at which we might be able to be doing all right, at least. And so the real question here for us as biodiversity scientists is how will this future climate change, the ongoing climate change and also the future, affect species, ecological communities, and biodiversity? And in concert with that, how will it affect the functions and services that they provide us? And so a lot of the work that Elise and I do here at MSU is to really try and understand how these changes are impacting biodiversity and species. And this is a really important area of research because understanding and quantifying the way that biodiversity is responding to these changes and how it will respond in the future, those are essential components to making decisions about whether a species is endangered or threatened. So for example, the IUCN, International Union for the Conservation of Nature, has a red list, which lists those species that are at highest risk of extinction. It also rates species that might be doing okay. And so to understand and inform those, we need to have robust analysis that can actually quantify those effects. Right here in the US, we have the Endangered Species Act, which is a similar type of designation. And collectively, a lot of this research can help us just understand where in the world we need to be identifying areas for conservation and designing reserves in particular. So for the rest of the talk, we're going to focus here on a couple of examples of biodiversity science going on at MSU. And we're going to highlight aspects coming out of each of our labs, but we want to make sure that you understand that there's a larger group of faculty and students and postdocs here at MSU involved in the Ecology, Evolutionary Biology, and Behavior program, or EEBB. And that's a cross-college program. It contains about 125 faculty, 101 grad students, and 27 postdocs. So it's a really large program. It's actually one of the largest in the United States, if not the world. And so there's a really strong component of biodiversity science going on here at MSU. And Elise is going to start by introducing the three vignettes that we're going to be talking about today.
Hi, everyone. So Phoebe, thanks for that great intro and overview of biodiversity. I think that sets up really nicely for the rest of the talk. So what we're going to do now is present three different vignettes. So the first one that I'm going to do is going to be talking about what's the factors affecting biodiversity decline, and I'm going to talk about a case study that we're doing with monarch butterflies. Then Phoebe's going to jump back in with the second vignette, talking about how we predict biodiversity across space and time from some of her research. And then we'll end with a third vignette talking about what are the consequences of biodiversity loss with the tropical amphibian and snake system that my lab has been working on. So next slide, please. Okay, so the first one I'm going to jump into is talking about the multi-seasonal stressors that have been impacting monarch butterflies. So monarchs are a system that my lab has been studying now for about 10 years. And I'm going to jump into why they're an interesting group to study, and also how it can shed light on many other organisms. Next. So monarchs are an extremely interesting species. And one thing that's maybe some of you don't know is the monarch butterfly east of the Rocky Mountains are all a single population. So they all intermix, and they have a really unique, interesting life cycle. They overwinter in central Mexico in these high elevation OML4 fir forests. Then at the end of winter, the early part of spring, so early March, they move up into Texas and the surrounding area where that first generation is produced. So those individuals produce that first generation. They go through an egg and larval stage and then metamorphose to adults. And then after that, that generation moves up into the Midwest, where they produce 2 to 3 more generations, Midwest and East too, and all the way up into southern Canada. And at the end of summer, early fall, the last generation, about the 4th generation, enters what's called reproductive diapause. So they kind of shut off their reproductive organs, and that last generation migrates all the way back down to those same high elevation OML4 forests in central Mexico. So this combination of this really unique migration and multi-generational migration to the same location, is unique among the world. There's really no other populations that we know do exactly that. And because they're so iconic, they're really easy to identify, and they're sort of ubiquitous, there happens to be quite a bit of data available from them, particularly from citizen scientists. So that presents a really unique opportunity to study what's going on with monarchs. Next slide, please. So unfortunately, the population has been declining from about the mid-90s. So this is a graph here that shows year on the x-axis and the area occupied in winter in those wintering colonies. So we can't count monarchs because there's hundreds of millions, but what we do is we delineate the amount of area in each of these separate little colonies that's been occupied. And this is in collaboration with the World Wildlife Fund in Mexico. And what's happened is the population has declined pretty precipitously since the mid-90s when they started collecting the data. And the decline has been almost about 90%. So they've rebounded slightly in the past several years, but still much lower than their previous numbers. And because of this, they're being considered for listing under the Endangered Species Act. So next slide, thanks. So what are some of the reasons for their decline? Well, one of them that people thought a lot is loss of habitat and overwintering mortality. So on the right here, you can see these figures. So there's one of, this is in the wintering habitat location. Here's one in 2013. There's the same area in 2015. And what you can see is that there's some illegal logging. Now, a reserve was set up in the mid-90s so that no longer allowed to log in the areas where these monarchs overwinter, but there's still a little bit of that. In addition, we've seen, as Phoebe mentioned, some climate impacts. So we've seen more storms in that area, ice events, and on the left there you can see mass mortality of monarchs on the wintering grounds. Next slide. So a big hypothesis for why monarchs are declining is a loss of their habitat and their resources of milkweed. So the adult monarchs lay eggs on milkweed plants. They have to, their eggs and larvae can only survive on it. After they hatch, the larval stages eat the milkweed. And what we've seen is a huge loss of milkweed in their summer habitats. And this is largely due to changing agricultural practices. So around the mid-90s or early 90s, many of you might be familiar with Roundup Ready and genetically modified seeds. So because of this, people kind of sprayed across large agricultural fields this glyphosate, Roundup Ready, and that led to a loss of a lot of milkweed, which used to grow in agricultural areas. So this graph on the right here shows the amount of glyphosate application, so the percent of crops in each of different counties that were sprayed from early 1990s to the middle 2015s or so. And what you can see is that we've reached almost about 100% or 90% on average now of these crops being sprayed. And we saw that kind of start happening around mid-aughts, so around 2004, 2005. Next slide, please. So another big reason is climate change. We know that monarchs take a certain amount of energy, what we call growing degree days, to develop from eggs all the way into adult individuals. And particularly, this becomes really important in that springtime period when that first generation that's pretty small is producing that first adults. So changes to the both precipitation in the Texas area and also temperature in that area have led to changes in the monarch population, which is something that we're working on a little bit. And then lastly, next slide, please. There's been some hypotheses out there that there's been an unsuccessful fall migration, so that might be one reason why the population is declining. And really the leading factors for this are two things. First, that there's been higher incidence of this disease, OE, which infects adult monarchs and makes their flying as unsuccessful, so they don't necessarily die as much, but they can't make the long distance migrations. And then the other reason has also been changes to the landscape and nectar resources, also related to climate. And then of course, potentially less habitat available once they get down to the overwintering sites so that they cannot successfully establish the colonies. Next slide. So one of the things of why this last hypothesis, this unsuccessful fall migration, has been really interesting is because of this chart here. So what I'm showing you is on the x-axis again is year, but on the y-axis, I have an index of peak summer population size of the monarchs. This is in the upper Midwest and southern Canada region. And what you can see over the same time period that we've seen monarchs declining in their overwintering ground, we've actually seen sort of a fairly stable index of this population in the summer, or at least we, from the best data that we have available from the citizen science data that we've seen. So because of this, this has really led to questions of looking at this fall migratory period, which is notoriously a difficult time to study, as you might guess, because the monarchs are moving vast distances over a really short time. So it's notoriously hard to study animals during migration, and this fall one is particularly hard. Next slide. So what we decided to do is to evaluate the potential drivers of population change during this fall migration. And this is work that was led by my postdoc, Sarah Saunders, and published last year in the Proceedings of the National Academy of Sciences, where we were trying to look at that fall migratory period. Now, we were lucky that we were able to study this period because there are so many citizen science data sets available. So this makes it a really great case study for looking at migration. So what we did is we looked at how much nectar available is there for monarchs during that fall season, during their migration down. We incorporated data on the disease, the annual amount of disease within the population, and also forage conditions at overwintering sites, how much core habitat and supplemental habitat was available at the sites and how that changed through time. Now I want to note that we looked at this from a time period of 2004 to 2015. And as you might remember, 2004, 2005 is about the time period when that glyphosate application, that spraying of herbicide, started to level off. So there's a lot of hypotheses that might be that spraying of Roundup Ready might have been one of the reasons that we saw the population decline in the 90s. But as that level saturated out, we're still seeing the population decline. So this time period in particular was kind of an interesting one to study. Next slide. So what we did is we included in our model two types of variables, these continental scale variables that were affecting monarchs during the migration, and these local scale variables at the overwintering habitat types. Next. So for the continental variables, we first divided the migration up into two different regions. Region 1, which is during the first half of the migration, and Region 2 during the second half of the migration. And we incorporated indices related to the temperature, the autumn temperature in each of those regions, and the greenness index, so how much nectar resources were available. We also included an effect of what was the peak summer population size. So we are wondering, if the peak population size is bigger in some years, does that mean that we also have bigger overwintering size? How much does that affect it? An index of the migratory roost, so how many populations and different groups that we see moving down from citizen science data, and also an index of the disease prevalence. Next, please. We included local variables at those overwintering habitat sites. So we include dynamics of the previous year. Even though it's all sort of aggregated as one colony, there's actually 19 different locations that monarchs overwinter in. So we said, okay, how big were those size colonies the previous year that might influence that? And were each of them occupied? Sometimes they're not, and they move around a little bit. How much forest and habitat available was at each of those different 19 locations, and then whether or not each of those sites was located inside or outside of this reserve protected area. Next. So what we found was really interesting. The first thing was that the largest variable that was having the biggest effect was that peak summer population size. So even though we don't see a decline in that in the summer index, the annual changes in the summer population size do have a big effect on what that winter population looks like. But we also found that the amount of greenness in the area, particularly during that first region, so at the initiation of the migration, was really important to how big the winter population size ended up being. And of course, there were lots of differences between that amount of forest habitat available. So we found a positive effect of that. And then not surprisingly, we found a positive effect of being located inside of the reserve. So areas that were protected and located inside the reserve generally had larger colonies. Next, please. So this research really highlights how much these effects going on throughout all the seasons and different time periods of the year, different geographic location, really influences the monarch population. So yes, the size of the summer population is really important, but it turns out that what's going on in the fall, the conditions during the fall, also affect how big that monarch population size. So right now, what we're trying to do is integrate all of the different seasons and time periods and factors that are available. Next slide. To try and look at the relative contribution of all these different stressors along the migratory route to the decline of monarch populations. And this is work that my postdoc, Aaron Zylistra, is currently doing now, where we're trying to quantify how much each of these stressors influence that dynamic and the decline of monarchs. So that wraps up my part. And now Phoebe, I think, is going to go into vignette 2.
Thanks, Elise. That was great. So I'm going to switch gears a little bit. And in this vignette 2, I'm going to talk about predicting different dimensions of biodiversity across space and time, which is sort of a key aspect of the work going on in my lab. So when we think about biodiversity research, it really spans a lot of different spatial and temporal scales, which Elise really highlighted well in that previous example. So a lot of the work that we do in my lab spans these scales as well. We work across space from local to global scales, across time from minutes to centuries. And these little images here are just showing an example of some of the current projects we have ongoing in my lab. And it's really important to span these different spatial scales collectively through using biodiversity science approaches, because at one end of the spectrum, we can perform experiments which actually help us understand the mechanism or the causes behind why certain organisms are responding in certain ways to climate change, for example. And those can then be connected up with the observed patterns that we see. And so these could be observed patterns at a global scale, taken through satellites, or through large observation networks. And I'm going to spend most of my time during this vignette talking about these larger scale patterns, but I want to make sure to just mention that KBS, the Kellogg Biological Station here at Michigan State, is really a world-class research station that allows us to ask these mechanistic questions, so reasons why certain organisms respond in certain ways to climate change in particular. And a lot of the work that we do in my lab focuses on this kind of central tenet, which is that warmer temperatures increase metabolic demands in ectotherms, especially insects. And that in turn causes them to increase their consumption rates. And so you can imagine that hungrier insects are going to have a larger impact potentially on the other species that they interact with, whether they're their prey or whether they're plants that they consume. And so these are just some images showing some of the experiments we have ongoing in both aquatic systems and terrestrial systems at Kellogg Biological Station, where we're actually manipulating temperature and precipitation, then also the composition of the species to try and understand how these effects of climate change are directly and also indirectly affecting those levels of biodiversity. And one of the central things that we look for is to try to identify, are there going to be certain species that respond more strongly and cause cascading effects across a whole ecological community? And the short answer to that is yes, there are some species that we're finding that are particularly sensitive to climate warming and that are also having a larger impact. And Elise is actually going to allude to some of these other larger impacts in the vignette she talks about next as well. So these interactions among species are really important. But before we kind of jump into looking at these patterns of biodiversity, I think it's important to understand where we actually get these data. And Elise alluded to some of this in her previous section. So we get these data across especially large geographic regions from scientific field surveys, from museums, and also from citizen scientists. And here's just an example of some of the different types of data sources that we use commonly in our research. I really want to emphasize that citizen science is especially becoming very important in the field of biodiversity science. Some of you may be familiar with iNaturalist or eBird, where you can actually participate and provide new information, which is then available to the larger scientific community. This map here shown at the bottom with all the red and yellow is showing just a screenshot of the available data that's offered through the Global Biodiversity Information Facility, or GBIF. And it's a collection of all these different data coming across the world, which we as scientists can then use to ask these fundamental and applied questions. So if we want to take those data, those observations that we see, and then just understand and map out patterns of biodiversity, there's actually a lot of science that goes into that as well. So I mentioned before that a lot of people think about diversity as being the number of species at a given place and time. And so you could actually describe that by what we call alpha diversity or site level diversity. And that's shown here just with an example of breeding birds in the United States. And so across the US, there's a survey that's done every year, and you can actually quantify and map the levels of diversity at a given site. And so sites might differ because they just have different composition of species. But let's say we wanted to compare differences between sites. We can actually come up with a different level of describing biodiversity patterns, and that's termed data diversity. So what's the difference between two or more sites? And as you can see, that map looks very different from the alpha diversity that we just created. Finally, we can add up all of the site level diversity for a given region and plot that as well. And that gives us what's called gamma diversity. And just looking at these three plots, they're very different. The maps show different patterns. And this is important because as conservation scientists are trying to preserve species across the world, using the metrics and being clear about those is an important area of research as well. So not only can we count up just the number of species, but we can look at how closely related they are. So phylogenetic diversity. We can also quantify their functions, how many functions they provide to the ecosystem. That's functional diversity. So as we fill out this whole matrix here of these maps, you can see that every image looks slightly different. And I just think this is an interesting way to actually show that there's so many different dimensions of diversity. And maybe an area to do more research in is to identify the areas that have the highest levels of all of these types of diversity. We can do a similar analysis with trees. Again, it looks different from birds. So in addition to providing some of these baseline maps of biodiversity, there's a lot of work in my lab that's working on trying to predict these patterns across space and time. But in order to do that, we actually have, relatively speaking, very few observation data points of these species compared with, let's say, a satellite remotely sensed image, which is detecting temperature across the surface of the Earth. And so we use the information about the environment through temperature, precipitation, land cover, and other variables collected by satellites to interpolate and describe those patterns of biodiversity. And the way this works is just an example here shown for these gray dots in Australia. This is a species that was collected with these locations. And then we can overlay those locations on these different layers from the satellite imagery. We can then come up with models that describe that relationship and apply that to identify the current and potentially, with climate change scenarios, future distribution of these species. And so there's a lot of work being done by many scientists across the world to generate these different models. And they're very essential, as I mentioned, for identifying reserve design and habitat that's especially vulnerable. But there's also a lot of issues with these models. They're fairly simplistic. They're basically relying on the fact that species and the environment have a singular relationship in many ways. And so one of the things that they're leaving out often is the unique spatial scales of what's actually driving patterns of diversity. We may see that climate has a pretty broad scale influence on patterns of diversity, whereas transitioning from an agricultural field to a forest or vice versa at a more local scale would have a local scale impact. And so understanding the scale at which these drivers impact patterns is really important for these models. Another area that is requiring a lot more work is adding in these different dimensions of diversity. The vast majority of these models are predicting taxonomic alpha diversity. And so there's so many more dimensions that we need to be investigating. And finally, interactions among species are largely ignored. They're fairly simplistic models that map a single species at a time. And these interactions are really essential. So in terms of understanding what drives actual patterns of diversity, excuse me, we are going to walk through a little example here. And one of the overarching patterns that we know from biogeography is called the latitudinal diversity gradient. So this image of the globe in the upper right here is showing areas of high diversity versus low diversity. And what you can see is high diversity tends to occur in the tropics, and low diversity tends to occur in the poles. And there's a variety of potential hypotheses for why this might be. One of the main ones is how much solar radiation is hitting the Earth's surface. It tends to be higher in the tropics, and that provides then essentially more productivity and more life for that area of the world. And so it also aligns with temperature. It just tends to be warmer in those regions, and the conditions, the environment, tend to be more amenable for higher diversity. But it's not really been shown from a lot of data that's been pulled through these different observation networks. And so we wanted to investigate this further. So the hypothesis is that temperature should drive patterns of diversity. We know from understanding the threats to biodiversity that this is not the single most important variable. Climate is very important, but there's a lot of other variables as well. There's multiple drivers, there's multiple spatial scales at which they act, and there's many interactions among them. And I'm going to just focus a little bit on two, so looking at the interplay between climate, temperature in particular, and what we term interspecific trait variation, which is essentially a way to describe the degree of competition or how important competition is among species within a certain community. And this is work that I've been working on with some postdocs at MSU. So to do this kind of research, we need a large observation network. We need a place that is collecting data in a very systematic way and providing all these observations. And fortunately, the recently funded National Ecological Observatory Network across the United States is a great example of this. And so here I'm just showing an image of what that network looks like. It's spread across these different climate domains and contains 81 sites. And at all these locations, the same type of data are collected. So data on different types of organisms and different types of environmental variables. At these sites, one of the types of data that the NEON scientists collect are on small mammals. And this data can help us answer this overarching question: why do cooler regions have fewer species? So here on the left is just an image, couple images showing the scientists actually collecting these data. They trap the small mammals throughout the summer, and then they weigh them. And those body size measurements, those mass measurements, can actually help us understand the level of competition in the community. If species have similar body sizes, they tend to be more competitive with each other, or they can compete more. And so over time, over evolutionary time, they might diverge and be less similar in terms of their body mass, just to avoid that competition, because competition is a stress. And so we developed a new overlap statistic to measure competition within an ecological community. And just on the top panel here is an image showing the map of diversity of small mammals across the United States, with the little circles showing where those NEON observation locations are. So in terms of this overlap measure, we could just look at the mean or the average body size of a given small mammal species, shown here on the left-hand panel. And that wouldn't really tell us much about competition, because we just know that one species is bigger than the other. But if we start to look at individuals and measure the distribution of body sizes within a species, we can get an idea of how much they overlap between each other. So in cases where we have a low amount of overlap of the body size between two species, that's evidence that those species have avoided competition and they're partitioning out their space and avoiding that. If we see high overlap, where all these species have a lot of overlap in their distributions, that's evidence that competition isn't as important, that they're able to coexist with each other, have similar body sizes. So lo and behold, we did this analysis across the National Ecological Observatory Network data, and we found that places like Wisconsin that are colder have evidence of competition being less important, because their body masses tend to overlap. This is an area where we see lower diversity of small mammals. So there's, in this case, 6 small mammal species in this colder region. That's in comparison with a place like New Mexico, which is hotter, and we've found that competition is more important. They have less overlap in their body mass, and which is evidence that the competition has been avoided over time. And so now it's basically partitioned out where they exist along this axis. And that helps explain why there's a higher number of species in warmer areas. So taken all together, this was a theory that was out there, and we helped to show through these data that actually warmer temperatures could reduce stressful cold environments, because cold is a stress, keeping warm is a stress, which in turn is promoting competition for resources in these warmer areas. So kind of going back to this complexity map and how we untangle what is actually driving patterns of biodiversity, I want to just end with a little section on geodiversity, which I think most people have not heard of as a term, and it's actually really important. So geodiversity is actually similar to biodiversity. It's the variation, instead of life on Earth, it's the variation of the abiotic or non-living components of Earth's surface. And it's really essential, because higher levels of geodiversity tend to promote higher levels of biodiversity, or at least that's a hypothesis. It hasn't really been quantified much. And so what we've been doing is using these NASA satellite remotely sensed imagery layers and actually computing different metrics of geodiversity and biodiversity to understand how geodiversity might be able to foster biodiversity. And what we found is that, yes, areas with higher geodiversity tend to have higher levels of biodiversity, but it's very dependent on where you are in the world, the different types of ecoregions, and it's very dependent on the spatial scale at which you think about any given driver of diversity, so climate, topography, land cover. And so this is some exciting research that's ongoing in my lab. So in summary, biodiversity patterns are really complex. Trying to figure out what is actually driving them is difficult. And some of the work that we're doing in my lab is helping to clarify that and providing some fundamental data products that other scientists can use. I'm going to turn it over to Elise now, who's going to talk about vignette number 3.
Thanks, Phoebe. That was really interesting. And I think it sets up nicely for the last thing that we want to talk about, which is what are cascading effects of biodiversity loss? So when we lose something, what happens to other taxonomic groups in that system? And I'm going to talk about a case study where we've been looking at what happens when amphibians are lost in system. And specifically, we're going to look at snakes here. Oh, sorry. Can you do the slide? I'm hitting it myself. So as you may have noticed in one of Phoebe's earlier slides, amphibians are one of the most threatened groups worldwide. So somewhere between 40 and 45% of amphibian species are threatened with both declines and some even with extinction. And one of the leading causes of amphibian loss, in addition to habitat loss and climate change like other species, is disease. And we've seen widespread disease that kills many, many types of amphibians. And there aren't that many diseases for other groups that are just that pervasive, that kill without regard to species. So that's kind of really disconcerting. Next slide, please. So disease of amphibians is actually globally distributed. And one of the most virulent is this fungal pathogen called BD, which causes this disease, chytridiomycosis. And really, because of this, many individual amphibians and many species of amphibians are impacted and eventually die by this. So greater than 500 amphibian populations have been extirpated worldwide. And you can see this map here kind of shows where this BD pathogen has been detected. And some of this lack of detection really is related more to surveillance rather than not necessarily being in these parts of Asia and Africa. You can see in most places where it's been looked for, it's been detected. Next slide, please. So I was interested in thinking about, okay, how are other taxonomic groups affected by this widespread loss of amphibians? So I worked on a project with 2 postdocs in my lab, Grace Dorenzo and Sam Rossman, along with collaborators at University of Maryland, to look specifically at how snakes are impacted by this loss of amphibians. And this is work that was published in Science earlier this year. Next slide. So why do we look at snakes? Well, many snake species rely on amphibians, mostly for prey. So you can see here examples of 2 different snake species that eat both eggs and adult phases. And the other thing about amphibians, and this is particularly true in tropical areas, is they're pretty abundant. And there aren't as many mammal species in these tropical areas where there's high diversity. So in certain regions, amphibians can be, frogs in particular, can be the main diet item of snakes. And in addition, because we've seen this widespread loss, not only do we affect this kind of direct impact related to the food source, but we think that there might also be other changes related to energy flow, because amphibians are both aquatic and terrestrial. You know, they have this tadpole stage and they also have an adult stage on land. And because of that, we think that there might be other kinds of indirect effects on snakes as well. Next slide, please. So our study area took place in this protected national park in Panama, right outside of the town of El Quepe in central Panama. And this is an example here of what you can see. So it's a really mountainous area, but it's also pretty secluded. So it was a good study area because there aren't other factors affecting this. The habitat has remained the same over the time period, which I'm going to talk to you the 14 years of our study. Because of that, the only thing that we really saw was kind of this disease change. So we could isolate this disease and look at how this loss of amphibians is affecting snakes, given that other things in that area haven't changed. Next slide. So this disease, BD, kind of swept through Central America. And my collaborator, Dr. Karen Lips, who's at University of Maryland, had been studying amphibians in Costa Rica when this BD pathogen came in. So she jumped down kind of to Panama, this area with the star in here, and was able to collect data for 7 years prior to this pathogen entering the area. Then in late 2004, chytrid BD came in, chytrid killed all of the amphibians, and Karen stayed in that area for another 6 years after collecting data. So she was mostly focused on documenting the loss of amphibians, but during that time, she also collected data on all the reptile species that she saw. Next. But the thing about it is that snake species in general, and particularly at El Quepe, are really, really rare. So we might have, for almost all the species there, you only see a few of them. They're occurring in really low abundance. And even some of the more abundant ones, they're not as abundant as lots of other taxonomic groups. So even when they're abundant, they're still much lower than other species, other kinds of species. And they're all pretty much difficult to detect. So this is an example on the right here where you can see, but even these ones that may look different, they don't move a lot, they're in dense forest habitat, and it's really hard to find them. And actually, because of this, 1 in 4 snake species worldwide has unknown conservation status. They're a notoriously hard group to study. Next slide. And here's an example, what you could see of where these transects, where these areas that Karen and her team were studying. There's actually a stream in there, and they're walking these really thick vegetative transects, collecting all data on information, every individual amphibian they see, what the species is, and every snake species. But you can kind of see probably how hard this surveying approach is. Next slide. So Karen and her team collected about 494 surveys before the arrival of chytrid, so before the amphibians were wiped out, and then just over 500 surveys immediately afterwards. But even with this huge number, over 1,000 surveys, the number of snakes that were observed was still really, really low. Next. So what we did was we changed our thinking. Instead of trying to estimate what's the absolute number of snake species in here, their absolute abundance or their occurrence rates, let's switch to thinking about, let's estimate the probability that the diversity and the occurrence of these species changed from pre to post the disease. We knew we weren't going to be able, because of this rarity issue and because of this low detection, we knew we wouldn't be able to estimate anything with abundance, with very high precision. Next slide. So I know we're running out of time, but I just want to wrap up and tell you some cool, interesting stuff. So basically we saw huge changes to the diversity of snake species. And I want to walk you through this slide here. So what we were trying to do is quantify the total number of species before, so that's in the blue, and after, that's in the orange, the loss of amphibians. So we call this species richness and the number of species. And so what I'm plotting here is the species richness on the x-axis and the probability of particular values that were generated by our model. So what we can find here in these dashed values on the bottom left is we can't have less than the number that were observed. So 30 snake species were observed before the loss of amphibians and 21 snake species were observed after. But what we find is there's actually a pretty high probability that there are many more there that we didn't detect. So you can see this 52.7 species before the loss of amphibians versus 40.1 species afterwards. And so we estimate that we may have lost between 12 or 13 species. And then what's interesting is this tail, and that is that there are really low probabilities. There's actually a huge number of species there that we just have never detected. So we can't say with certainty exactly how many species were there before and after, but what we can find is, next slide, this 85% probability that there are less snake species there now than there were prior to the loss of amphibians. Next. So we're doing the same kind of thing with the individual species. So I'm plotting here the species with at least, the 17 species with at least 5 total detections. And what we did here is the same kind of thing. What's the probability that their occurrence rates in our studying area are lower now than they were prior to the loss of amphibians, taking into account that we know that we're not detecting them perfectly and everything. So next, so the areas that are in red, so these 9 species that are circled red, had a decreased occurrence probability. So 9 out of the 17 had lower probabilities of occurring or lower occurrence rates in our study area. What's also interesting is that we found a few species, 4, that had increased rates. So while most species decline, a few species actually increased, and that leads to what we call biotic homogenization, which is everything becoming more similar on a sort of regional level. Next. Same sort of thing when we looked at the body condition of the individual snake species. So we had even less data on individual levels, but there were 6 species that had a sufficient number of samples both before and after the loss of amphibians. And what we find is those 4 on the left had high probabilities that they had reduced body condition overall for the species. But a few, like the 2 on the right, had actually better body conditions after the loss of amphibians. So same idea, most species do worse and a few sort of do better. Next. So just to wrap this up here is this research really highlights that when we lose something in a system, it can have really big cascading effects on other species. So our research was interesting because it focused on snakes, which are sort of at a higher trophic level than amphibians. But other research in this area has shown that things that are on lower trophic levels, like so invertebrates and primary producers, have also experienced changes as a result of the loss of amphibians. And what I also really want to note here is that this is notoriously difficult to quantify for a lot of different reasons. First is you have to know when some sort of loss event is going to occur. And a lot of times we don't know that a priori. So in this case, we were lucky to have been able to team up with Karen Lipson, where she had had data collection for 7 years before the loss of amphibians. But a lot of times we don't know when we're going to lose something. So we don't have that baseline data. And then the other thing that's really difficult is many, many species, particularly in areas of high diversity, like in the tropics, the species occur in lower abundance, right? So it can be difficult, and they can be difficult to detect. And because of that, it can be really hard to say with certainty what's going on. So these other species here that are highlighted in white, sort of the lizards, the fish, small mammals, birds, other species in this area might also be affected by amphibian loss, but we don't have any data on that. So those are going to be things that we never know.